Good afternoon, everybody, and welcome to our symposium. My name is Michael Niederman, and I'm from New York, and I'll be moderating our symposium today, which is devoted to the treatment of multi-drug resistant pathogens causing ventilator-associated pneumonia. I think we're all very familiar with the challenges in dealing with these difficult-to-treat pathogens in patients with pneumonia, particularly in the ICU. And for a long time, we've had very few treatment options available for us in dealing with the most difficult pathogens. Today we're going to explore three different avenues for improving the therapy of MDR pathogens. I'm going to first talk about some new antibiotic developments specific to ventilator-associated pneumonia. Carlos Luna from Buenos Aires is going to discuss the potential of using aerosolized antibiotics to treat ventilator-associated pneumonia. And Jean Chast from Paris is going to discuss non-antibiotic therapies for pneumonia. And I guess in a general way, this symposium is good news. It's good news because we are talking about something that we haven't had in the past, which is options that may be emerging for the treatment of difficult pathogens. And so in that regard, it's good news. It's also, I think, good news because this symposium is sponsored by the Academy of Infection Management. And this has been an organization that's existed for many years. It's been on a hiatus recently, but it's been very much committed to education of physicians and healthcare providers in the treatment of difficult infections, particularly focused in the intensive care unit. So we're very excited to be able to do this symposium and to do it on behalf of AIM, as I think this organization is going to be able to provide education on an important topic in the future. So I'm going to be talking about new antibiotic developments for ventilator-associated pneumonia. And the question is, can these new antibiotic developments help us in the intensive care unit? I've listed here my potential conflicts of interest. And again, I think it's interesting to get the historical perspective on where we stand with antibiotics. We have very bad bugs, and it's not what it was when antibiotics were first developed in Fleming's era. This was a list of his sensitive and resistant pathogens, and if you look at the organisms that he was concerned about in the 1940s, it was nothing like the difficult pathogens we deal with today. We have increasingly drug-resistant pathogens. The escape pathogens, I think, may be known to all of you. Not all of these organisms cause pneumonia, but escape is a algorithm that stands for Enterococcus faecium, which is VRE, Staph aureus, Klebsiella, E. coli, and ESBL producing organisms, as well as those who produce carbapenemases, Acinetobacter, Pseudomonas aeruginosa with multi-drug resistance, and the Enterobacteriaceae. And of course, all of the organisms shown in yellow here are the gram-negative pathogens, which we're particularly concerned about causing pneumonia. Staph aureus is an important cause of pneumonia, but of course a gram-positive organism. And of course, every year things get worse. This is one of the earlier reports about the metallo-beta-lactamases and the New Delhi beta-lactamases, which again are highly resistant to almost all classes of antibiotics. So why is resistance such a problem? And the answer is, of course, we're using too many antibiotics. I don't know how many of you are familiar with Pogo, cartoon character, but I think he said it very well. He said, "We have met the enemy and he is us." We are the problem. We are responsible for antibiotic resistance because we overuse antibiotics. And there's certainly been a renewed interest, of course, in the United States with antimicrobial stewardship, but I think around the world there's been great interest in avoiding overuse of antibiotics so we can preserve what we have. And as you may know, the Infectious Disease Society of America developed their 10 by 20 campaign under the guise of bad bugs need drugs. They wanted to develop 10 new antibiotics by 2020. And I would say realistically, that is likely to have happened. I think that we have a number of drugs, as I'll show you, in development, and it's promising that we might be able to use some of these agents in the intensive care unit. There's a lot of reasons, though, that we have not had drugs developed for a long time, and I think that some of it is regulatory and some of it is financial. And I think it's important to look at the financial implications of antibiotics, because because I think it's becoming less and less interesting for pharmaceutical companies to develop new antibiotics. For example, they could develop a new antibiotic at a tremendous development cost, and it would not cost them any different to develop a new drug for musculoskeletal disease, diabetes, hypertension. Those drugs, if a patient starts taking them, they may take them for years. Antibiotics are very short term, and although there are many more people who might need antibiotics, the profit margin on antibiotics is probably less than some of the other drugs that they could develop for the same cost. As I mentioned, there's a great interest in stewardship, and all of the guidelines now are focusing on shorter courses of antibiotics, and that's really in contrast to chronic use of drugs that we see in chronic disease. And so the most that probably a drug could be reimbursed is $3,000 per case, and again, if they develop a new chemotherapy drug, they might get $60,000 for a single treatment with some of the newer chemotherapy agents. So there's a number of financial reasons that pharmaceutical companies have not been particularly interested in developing new antibiotics. And again, if you're interested in this, I won't belabor this. There's a very nice review in the Blue Journal several years ago that talks about why antibiotic development is so slow and what some of the solutions might be. But again, I think we're only seeing some of that happening, and you're going to hear about some of these solutions today. On the other hand, I think we're aware of the problem with new drugs, and even as early as in the 40s when Alexander Fleming, in 1945, who had been the Nobel Prize winner, as you know, for developing penicillin, in his Nobel Prize lecture said, "There is the danger that the ignorant man may easily underdose himself and by exposing his microbes to non-lethal quantities of the drug, make them resistant." So I think we were aware a long time ago of the potential of these new drugs to not necessarily work indefinitely, and resistance has always been in the background. There's an unmet need for new antibiotics. This was a review of the Emergent Infection Network members. Over 500 people responded, and they rated their unmet needs for treatment of infection on a scale from 1 to 5, and MDR gram negatives were the number one unmet need. New antibiotic development they thought was the best strategy to develop or to meet this unmet need. So there's no question that everybody perceives the need to develop these new antibiotics. There are a number of drugs now available for gram-positives that are being developed. The oxazolidinone class is active against linezolid-resistant MRSA. The oxazolidinone class, of course, includes linezolid, but in development now is tadazolid, which has lower MICs than linezolid against MRSA. It has once daily dosing, no serotonergic agent interactions. Skin and soft tissue infection trials are done, and there are ongoing studies now in healthcare-associated pneumonia and ventilator-associated pneumonia. Ridazolid concentrates in macrophages and neutrophils and is another agent in the class that's being developed. The glycopeptide cephalosporin heterodimer that really only has a number now is active against gram-positive infections, including VAP with bacteremia. There's a phase 1 trial that's now being conducted with this agent. In the tetracyclines, we have omadacycline, which is both IV and oral, primarily used for community-acquired pneumonia, but does have activity against Staph aureus and some gram-negatives. And so many people have sort of anticipated that this is similar to an oral version of tigecycline with both an oral and an IV option, and we may see this be developed for the treatment of ventilator-associated pneumonia as well. Tadazolid versus linezolid have been compared, and again, tadazolid is a little bit more active, particularly against MRSA than linezolid. Again, I apologize for these MIC slides. I don't really like reading them, but I flashed them up here to show you the references if you want to look a little bit more closely at some of the activity of individual drugs. There are a number of drugs now that are being developed for gram-negative VAP. Those include ceftolozane tazobactam, ceftazidime avibactam. I think these two beta-lactam, beta-lactamase inhibitors are the ones that we probably ought to be spending the most time thinking about. Both of them have approval. One of them, ceftaz-avibactam, is approved in Europe now for pneumonia. Neither of them are approved for pneumonia in the United States. There's an aztreonam-avibactam, which is active against metallo-beta-lactamases. There's a siderofluorocephalosporin being developed. There's carbavance. There's a meropenem with like agent, which is with an inhibitor called vaborbactam, which has enhanced activity against KPC-producing organisms. Imipenem relebactam is being developed. Plasmomycin, which is like an aminoglycoside. There's a new macrolide inhibitor, and there's a cyclopeptide. And again, all of these may sound unfamiliar to you, which to me is very good. It's very good because it means that we really do have antibiotics being developed in different classes than we're familiar with, and all of which have the potential to help us in very difficult situations. I think the biggest development, however, in new antibiotics has been the new beta-lactamase inhibitor. And there are now 3 beta-lactamase inhibitors. There's avibactam, there's relebactam, and then there's vaborbactam, which is the one that's got the numbers here. And again, if you look at them, they're all good against Class A narrow-spectrum beta-lactamases. They're all good against Class A ESBLs. But against the carbapenemases, not tazobactam, but all 3 of the new ones, some Class C beta-lactamases, all are active again. And avibactam is the only one that's active against Class D beta-lactamase producing enzymes. And so these are all new developments that potentially can help us. And I've developed this list here that talks about new drugs in the pipeline for resistant gram-negatives. Ceftolozane tazobactam is approved now, but not for the treatment of nosocomial pneumonia. Ceftolozane is a new beta-lactam that's got enhanced pseudomonal activity over the previous beta-lactams. It's with a very familiar beta-lactamase inhibitor, and there are ongoing trials now in ventilator-associated pneumonia. Ceftaz-avibactam has been a very familiar beta-lactam, but a new beta-lactamase inhibitor, which, as I pointed out on the previous slide, is probably the one that's most active against all the different classes of beta-lactamases. And again, it's got good pseudomonal activity as well. Ceftaroline-avibactam is primarily an MRSA drug, and again, what role it'll play in VAP is to be determined. The new cephalosporin beta-lactamase inhibitors are only available intravenously. Ceftolozane tazobactam and ceftaz-avibactam, again, are active against Pseudomonas. Ceftaz-avibactam is active against a very broad range of KPC-producing organisms, and tazobactam may be different from the avibactam because tazobactam has irreversible binding, whereas avibactam is reversible and can be recycled, so that potentially avibactam could recycle after the organism is killed. If we look at the activity against these new drugs, again, ceftolozane tazobactam and ceftaz-avibactam have very similar spectrums of action. Again, probably ceftolozane is a little bit more active as an anti-pseudomonal agent, but again, ceftaz-avibactam is also a very good pseudomonal agent as well. I won't go through again the MIC data, but the references here if you want to look closely at the in vitro activity of ceftolozane tazobactam and ceftaz-avibactam. Those numbers are published. And if we compare these two drugs to one another, again, ceftaz-avibactam is active against KPCs and ESBL. The dosing is 2,000 milligrams of ceftaz with 500 milligrams of avibactam, so a 4-to-1 ratio. It has predictable pharmacokinetics, rapid drug distribution, and renal excretion. Ceftolozane avibactam is extremely active, again, against ceftolozane tazobactam is extremely active against pseudomonal resistance mechanisms. It's dosed in a 2-to-1 ratio, 2,000 milligrams of ceftaz, 1 of of ceftolozane, 1,000 milligrams of tazobactam. Now, in the United States, this dose, which is referred to as a 3-gram dose, is not approved for the treatment of pneumonia. The dose that's approved for skin and soft tissue infections is the 1,500. And the trials that are ongoing now in the treatment of ventilator-associated pneumonia use this higher dose of 2,000 milligrams of ceftolozane, 1,000 of tazobactam. So I think if you're going to use it off-label to treat VAP, you need to use that higher dose that, again, is not approved in the United States. It's active against ESBLs, again, also predictable pharmacokinetics with rapid lung distribution and renal excretion, but it doesn't have KPC activity, whereas avibactam, again, is active against KPCs. Again, the dosing we mentioned, but again, if you're going to use ceftolozane tazobactam, it's important to give the 2-gram dose. When you're using ceftaz-avibactam, 2 grams plus 1, so the 3-gram total dose. When you're using ceftaz-avibactam, you can administer it as a 2-hour infusion, which I think is really an advantage for targeting potentially more resistant pathogens. Again, this is a distribution of target attainment in epithelial lining fluid following the highest dose of ceftolozane tazobactam. And again, if you want to target the most resistant pathogens, you need to use that 3-gram dose. The 1.5-gram dose, which is approved but not for pneumonia either, doesn't target nearly as resistant organisms as the higher dose. So again, if you're going to use it before all the trials are done to treat VAP, I think most people would recommend you use the 3-gram dose, which is again 2 of ceftolozane and 1 of tazobactam. A couple of other drugs to think about. Relebactam is imipenem relebactam, which is another beta-lactamase inhibitor, active against KPCs. Plasmomycin is called a neoglycoside. It's very much like the aminoglycosides. And brilicitin is a peptide defensin protein, a totally different type of mechanism of action than some of the cell wall antibiotics we've been talking about. So if you go to clinicaltrials.gov, you can get updated on where we stand with some of these drugs in VAP trials. Ceftolozane tazobactam in the 3-gram dose is being compared to meropenem for nosocomial pneumonia. This is a drug that's been developed by Merck. The endpoint here is all-cause mortality in a little over 700 patients, and the trial is still ongoing. Imipenem relebactam, which is another Merck product, is being compared to pip-tazobactam for hospital-acquired pneumonia, including VAP, and again, they're looking at 28-day survival. And tadalafil, again, another Merck drug, is being looked at the 200 milligram dose a day, daily versus linezolid 600 milligrams twice a day, and again, all-cause mortality at 28 days is the endpoint. Now, it's very interesting to think about the way that these trials have been designed, because Merck has been very much attentive to the US FDA recommendation that all of these drugs have to show non-inferiority for mortality. So in all of their trials, their endpoint is mortality and demonstrating non-inferiority. That's not necessarily the case for some of the other drugs. Plasmomycin is being tested with adjunctive antibiotics, also looking at all-cause mortality. But if we look at the ceftaz-avibactam trial, it's being compared to meropenem. It's including VAP and non-ventilated pneumonia. And here the primary endpoint is clinical cure. And this trial has been completed, and it's the basis of the drug approval in Europe. But I think there's some serious discussion as to whether or not this is going to present a problem in the United States, because what's potentially problematic in the United States for approval is they didn't use as their endpoint all-cause mortality, and they also mixed VAP and non-VAP. So I think when we're looking for which drugs will be approved for VAP, I don't think we know if this trial design will or won't present a problem. If you actually go to the website and read about what they've done, they've enrolled 356 patients who got ceftaz-avibactam, 370 who got meropenem, Again, the clinical cure rates were, again, quite similar, and there was a slightly better p-value for clinical cure with the ceftaz-avibactam. They got similar data for test of cure at the end of therapy, but again, no comment here about all-cause mortality. So I think what the only editorial comment I'll try to leave you with is if we're really going to use these drugs to improve ventilator-associated pneumonia management, I don't know if these trial designs are really going to do it for us. Because all of these trial designs, no matter whether the endpoint is clinical cure or mortality, are designed to show non-inferiority, and they generally require that the pathogens be sensitive to both the new drug and the old drug. So in other words, by design, none of these trials will ever show whether or not these new drugs could help us with our most difficult to treat pathogens that can't already be treated by existing drugs. Because to get into any of these clinical trials, the organisms have to be sensitive to an existing drug. And I think what might be better is that we design for superiority against the best available therapy using the new agent and using highly resistant pathogens. Take pathogens that we probably couldn't treat today and treat them the best way you could without these new agents, and use these new agents with maybe the best available therapy as well, and see if there's truly superiority. And again, I've turned to a very famous quote from the past: every system is perfectly designed to get the results it gets. We have trials that are perfectly designed to not answer the question, will these drugs work in situations that our current drugs don't work? They're not designed to answer that question, and that's the only comment editorially that I would say we really need to have that question answered. So to conclude, there are some new agents in development and being tested in clinical trials. There may be new therapies for VAP, both IV and aerosol, which Carlos is going to talk about, but the trials are designed for equivalence, and to me that's a potential problem. Most of these drugs are modifications of older agents or with the addition of a new beta-lactamase inhibitor, but there are some new classes. There are pleuromutilins, there's the sideroflor, can't pronounce it right, the monosulfactam, the cyclopeptides, and the peptide defensin protein mimics. And again, I think the other question that many people have talked about, which to me is not really the answer we need, is if we're going to design these trials to show equivalence and not superiority, then at the maybe what they can help, these new agents can help us with, is stewardship. Maybe we could use them in place of carbapenem and make them carbapenem sparing agents, but I'm not sure that it really has been worth all this effort to develop new agents only to be told we can't prove that they're any better than the old agents, so let's use them instead of the old agents to spare some of these agents so that they too will remain active for a long time. So my only real concern is we have a number of exciting new drugs, but I'd like to see the trials be better designed to really test the potential of these drugs to help us in situations where we just don't have answers today. Thank you very much. So I'm going to move on to our next speaker, which is Carlos Luna from Buenos Aires, and he's going to be discussing the use of aerosolized antibiotics for VAP.
Well, thank you very much. Thank you very much, Mike, and thanks for this invitation to speak for AIM that is supported also by me. This is a group of, this is like that something happened more than 20 years ago, and we're still talking together about ventilator-associated pneumonia. At that time, we were interested in the diagnosis and in the use of appropriate therapy, and now we know that we will not have antibiotics soon, and this is now our concern. Well, this is my financial disclosure, and well, I will speak about the aerosolized antibiotics. And the story of aerosolized antibiotic is older than us. This happened more than 40 years older than us as speakers, at least. 40 years ago, we could find papers talking about the local installation of antibiotics in people in the ICU. And they were looking not exactly on infection or colonization, but there were a lot of studies made at that time. About in the 90s, they appeared some studies made with nebulizing antimicrobial, but nebulized not with the newer nebulizer, but by the usual nebulizer used, for example, for the administration of bronchodilators. The problem with the antibiotics, with the susceptibility, with the high levels of resistance, had reborn the interest on the nebulized antibiotics. But there are some considerations that have to be taken into account, because these nebulized antibiotics is the pharmacokinetic, pharmacodynamic, and the importance of this is different than what happened with the nebulization of bronchodilators, for example, in asthma or in COPD, where this way to administer the therapies is usual. For antibiotics, it is necessary to know the precise dose necessary to balance efficacy and side effects. The cost of the drugs is much higher, and what is very important is that we have to minimize the environmental exposure of with this drug, because it's harmful for everybody where the patient are. This is from 10 years ago, when this paper was written, as the different brands of manufacturers for the different nebulizers. And this is because there was the Hess et al. Did a study evaluating how was the efficacy of the nebulizer to give an output of the drug, and how much of this drug was available in the airway of the patient. And we can see that there is a big difference in the output of the nebulizer, and how much is available with the different trademarks of nebulizer. So this is one of the things important. And the other thing is the difference between the different nebulizers. So the nebulizer must be able to produce a very small particle, approximately 7 to 5 microns, and to be able to achieve the alveoli, that is where the pneumonia is and where need to achieve. There are several issues that have to be taken into account. Related to the ventilator, the mode, and how is the different issues on the ventilator. Related to the device used to provide antibiotic, is not the same metered dose than nebulizer. The issues about the drug, the dose, the size of the particle, the duration of the action, related with the ventilator, with the circuit, with the endotracheal tube, and the gas humidity and density. Also related with the patient, with the severity of obstruction, with mechanics of airway obstruction, and presence of organic, or the dynamic hyperinflation, and the synchrony. So, but there is a lot of issues for all of you now about the severity of HAP and VAP, particularly VAP, that is a disease that have a high mortality and a high attributable mortality also. And the gram-negative pathogens, particularly the multi-drug resistant pathogens, account of 65% or more of the cases of pneumonia in the intensive care. And the fact that also that the aminoglycosides are suitable. The problem is that when administered intravenously, they have not very good PK and PD. So the administration of this therapy by nebulizations could achieve, yes, very high concentration and very useful for therapy. So we require a treatment with antibiotic pneumonia at high concentrations in the lung where they have to act. And there are issues with toxicity also, and this could be achieved by nebulization without producing high levels of antibiotics in blood. Let me see. Go on. There have been a lot of research about this before applying in the humans, and one of the way to analyze this was the piglets model, developed by the people in France, by Charles Hugo Marquet and Ruby and so on, and they more than 10 years ago, so 15 years ago, they developed or they participated in the development of this new model of nebulizer that act using a piezoelectric transducer and vibrating waves, and a huge number of vibrations per second, something like 100,000 of cycles per second, as make the very small particles of adequate and they have no air added to the system, so very convenient to obtain a very good concentration of the antibiotic in the inhaled air during the mechanical ventilation. And the concentrations that are achieved in the lung tissue in the piglets that were inoculated with pathogens, gram-negatives, is very high. Here you can see what is the concentration obtained in the lung tissue, and this is in the concentration obtained in the lung tissue when it's given by intravenous, as usual. And this is 5 microgram per gram, and this is 180, so is about 20 or 30 or 40 times more than which is the concentration obtained given by intravenously. And the same happen, depends if the pneumonia is severe, the air that achieve the lungs is less, so it's not so good, but the concentration even here is 10 times more than they obtained by intravenous administration. So this makes that the antibiotic administered in this way are very highly available in this lung. This was, this group and other groups studied different antibiotic, and the result was more or less the same. This is another study made for by the same group. I have been there and I took this photograph. This was in 2002, 15 years ago, and they were working at that time with ceftazidime. But again, this system may be very closer to avoid the contamination of the air where everybody of us were, and this is one more of the difficulties to obtain this kind of nebulizations. And again, intravenous administration achieve a concentration here, very small, was higher with the nebulization. And if the nebulization was done with the nebulization with helium, the because of the lower viscosity was a little higher too. This was in the study by substacity. Again, more or less the same of the other figures is in another study by Gorsen of the same group, looking at which was the in micrograms per gram after in the autopsy of the pigs, the concentration of the antibiotics when was given by aerosol versus intravenously in different lung segments. And here, interestingly, in the piglets that are ventilated in a prone position, the concentration is lower than in the rest of the lung, but again, the concentration is several times more than the concentration that is achieved when it's administered by intravenous way. Again, the comparison, we have seen this slide before, and this is the efficiency of the deposition and in healthy piglets and inoculated. The inoculation made the appearance of pneumonia reduce, but not so much the concentration in the lung in the autopsy, but again is much more higher than the concentration obtained when it's given by intravenous. Here we can see when this is the blood concentration when this administered in the closed circles when it's administered intravenously, and this is the blood concentration when it's administered. This is nebulized intravenously, and we know that as is expected, the blood concentration is less than half in the peak of the concentration, but then obtain a higher concentration than when it's administered by intravenous. That is in the blood concentration. And they observed and obtained several conclusions about the lung concentration of amikacin between 3 and 30 times higher than the obtained when it's administered intravenously. What I said is that the antibiotic deposition in the infected lung and the bacterial efficiency were better with the nebulization than when it's administered intravenously, but have to say to keep in mind that the presence of pneumonia make to attain a lower concentration even using nebulization. The PDDS is nebulating vibrating mesh nebulizer that is estimated that deliver 45% of the administered dose to the lungs, and has other properties. The size of the droplets is 3 to 5 microns, and the controller of the nebulizer makes that the aerosol is delivered only during the first 75% of the inspiratory phase, making that the amount of antibiotic nebulized is about 45% of the total, much more than with any other kind of nebulizer. This is the PDDS nebulizer. This was used in a study of the pharmacokinetics by the group of Jean and Louis et al. Was published some years ago, and they observed that the PDDS delivered aerosolized amikacin with a very high amikacin concentration in the epithelial lining fluid, and safe serum amikacin concentrations. Here we can see the amikacin concentration in the different areas, and in this other study that was published in Intensive Care Medicine was a multi-center randomized placebo control. Mike Niederman was the first author, and they made the randomization of 69% who received standard antibiotic therapy plus the amikacin nebulizer, and the one received placebo, the other one received placebo in one of the doses, and the other one received 2 times a day the nebulization with 400 milligrams of amikacin. And the primary point was the tracheal aspirated amikacin maximum concentration more than 6,400 micrograms per ml, and the ratio of area under the aspirated concentration curve more or the same as 100 on the first day. This is from other study, but looking at what is the difference of we have seen this slide previously. The mean amikacin plasma concentration when it administered by aerosol, that the closet and intravenously, and another study that was published by Niederman was presented in the ISICEM in 2007, observed the difference what happened when the patient received the nebulized amikacin in different doses, and they observed that the number of antibiotic used by the end of the therapy was less with the twice a day amikacin compared with the once a day, and compared with no amikacin received as in any time. There is another study that recently finished that was a comparison of the use of aerosolized amikacin plus fosfomycin delivered via a flow inline nebulizer, also a vibrating nebulizer. Is a phase 1 study evaluating the PK and safety tolerability of amikacin fosfomycin using a vibrating plate nebulizer. And in this study, the 9 patients in this precise study, they looked at just the use of different doses, and what they observed at the day 3, when they received 2 blinded randomized treatments, amikacin fosfomycin and a placebo. What they observed is that there was a high tracheal aspirated concentrations of amikacin and fosfomycin, and the airway clearance was rapid, and no adverse respiratory effects were noted. So the administration of nebulized antibiotics is one of the ways that we can try to fight against the infections that so resistant to antibiotic, and this high concentration obtained with the nebulization is one of the ways that is used trying to have success in this fight against the resistance. This is the nebulizer used by Montgomery et al. Summary is inhaled antibiotic as adjunct to systemic antibiotic show promise. Aerosol delivery to ventilated patients is associated with high drug losses, poor reproducibility, and deliver a low have a low delivery efficiency, but vibrating mesh nebulizer for specially formulated antibiotics yield high local concentration and rapid clearance and low systemic exposure. It is necessary to do large multi-center trials, but to determine if this would be translated in improved clinical activity and decrease of microbial resistance in BAP patients. And well, it is needed to develop and optimize the use of aerosolized antibiotic. Thank you.
Our last speaker is John Costa, who's going to discuss non-antibiotic therapies for pneumonia, but really focusing on the potential of other new therapies for MDR pathogens.
So good afternoon, everybody. Thank you, Mike, for the introduction. So this is my disclosure, of course. As you know, as ICU doctors, when we are facing a severe bacterial infection in the ICU, our goal is very simple: killing as soon as possible, as much as possible, the microorganisms responsible for the infection. This is a Terminator strategy, of course. But unfortunately, the bugs have a lot of mechanisms to resist to the action of antibiotics, going from blocking the penetration of the drug inside the bacterium to extruding very rapidly the antibiotics from the inside to the outside of the cell. And it's also possible, of course, for the microorganisms to modify the target of the antimicrobial agent inside the bug, or to destroy the antibiotics. For example, using a beta-lactamase for many antibiotics, as you know. Of course, we are responsible, as said by Michael, by the selection pressure we are doing, giving a lot of antibiotics to our patient for the emergence and dissemination of resistance to antibiotics. But remember, we didn't invent the majority of antibiotics. The antibiotics themselves were in fact invented by the microorganisms themselves. And this is probably the most well-known example of that. As you know, Fleming didn't invent penicillin. In fact, okay. Thank you. In fact, penicillin was invented by a fungus, which is named now Penicillium, and of course, this is the true inventor of that antibiotics. And most antibiotics we are using were in fact invented by microorganisms. But the microorganisms are not crazy at all. At the same time, they invented the new antibiotics. They also invented the mechanisms for protecting themselves from the new antibiotics. Because, of course, the fungus producing penicillin should be able to resist to penicillin, not to be killed by the penicillin, of course. And we have, in fact, very strong data showing that, digging very deeply into the permafrost in Alaska, it was possible to recover deep in the ground the microorganisms present more than 30,000 years ago, a long time ago, before any human beings capable of inventing an antibiotic, of course. And interestingly, using metagenomic techniques, it was possible to find inside these very old microorganisms the element responsible for protecting the bugs from vancomycin. The bugs invented vancomycin more than 30,000 years ago, but also the mechanisms protecting themselves from vancomycin. This is a very interesting study, I think. But moreover, even using very effective antibiotics, particularly in patients infected into the lung, it's very difficult to eradicate the bugs from the airways, as you know. This is a very old study done in the UK, demonstrating that just after introducing the new antibiotics for treating patients with a proven microbiologically documented pneumonia, that okay, it was possible to obtain, using antibiotics, a small reduction in the number of microorganisms into the proximal airways. But after a few days, still, there are a large number of bacteria still present into the proximal airway secretions. More than 10 to the 5th colony forming unit by ml. So, okay, it's possible to decrease the bacterial burden into the proximal airways using antibiotics, but only during a small period of time, maybe. And we have also the data showing that. This is a study we did a couple of years ago, comparing 2 durations of therapy in patients with ventilator-associated pneumonia. As you can see, even using a pretty long duration of therapy, 2 weeks of treatment, first of all, the mortality was still pretty important, but also we did observe a lot of relapse, despite, in most of the patients, a regimen combining at least 2 antibiotics efficient against the bugs. So really, we need to be able maybe to modify our strategy. And maybe we need to go from the Terminator strategy to the Gandhi strategy, a non-violent strategy. Using such a strategy, we are now targeting the virulent factors used by the bacteria to target the host, okay? And we have now a lot of new targets for making the bugs completely inoffensive. But we are not anymore killing the bugs, which is a major difference. And there are a lot of potential, of course, advantages doing that. First, we have good data showing that could be effective, efficient, in fact. As you know, Emil von Behring was awarded the first Nobel Prize in medicine a long time ago for his discovery of the diphtheria antitoxin. This is exactly the same strategy, in fact. But there are also some other potential advantages for such a strategy. For example, this will maybe have increased efficacy. We don't know, but it's a possibility. It will also support antibiotic stewardship. And very importantly, it could be able to use this strategy not only for therapy, but also for prophylaxis, prevention of infection, or a preemptive strategy, which could be interesting also. And we have some proof of efficacy of such a strategy, for example, regarding Clostridium difficile infection. As you know, particularly in older patients, it's very difficult to avoid the recurrence of the disease, whatever the antimicrobial agent you will use. But using a monoclonal antibody targeting the toxin produced by the bugs, it's possible to reduce the recurrence rate observed, as shown on the slide. Another possibility for this non-antibiotic approach would be to target the quorum sensing system. As you know, this system is used by many gram-negative bacilli, including Pseudomonas aeruginosa, and this system permits to the bugs to monitor their own population densities and also to activate some virulent genes. In other words, going from harmless type to a very aggressive type, okay? And this is a major mechanism used by many gram-negative bacilli for targeting human beings. And as you may also know, it's possible to interfere with the quorum sensing system using macrolides. Macrolides have practically no activity against gram-negative bacilli, but some macrolides, such as clarithromycin, could interfere with the quorum sensing system and therefore modulate the density of the population of the bugs. And those Greek investigators did a randomized control study in patients with VAP, and patients were randomized either to a conventional strategy or to a conventional strategy plus clarithromycin for 3 days. And interestingly, in that preliminary randomized control trial, it was possible to document at least a benefit regarding the duration of mechanical ventilation, as shown on the slide. It's also possible now to use monoclonal antibodies specifically designed to target one or several factors of virulence. For example, as shown before, as indicated before, for neutralizing the toxin produced by the bug or targeting the aggregation of the bugs. So many possibilities for interfering with the virulent factors used by the microorganisms. And it's using monoclonal antibodies, for example, it's possible to target the type 3 secretion system, which is a major virulent factor used by gram-negative bacilli and, for example, Pseudomonas aeruginosa. As you know, the type 3 secretion system involves a needle-like complex that penetrates the host cells for injecting inside those cells toxins. And it's possible to use a monoclonal antibody to make the type 3 secretion system ineffective, blocking the needle and the injection by that needle of the toxins. So, and we have now good data in animal models showing the efficacy of such a strategy, blocking the type 3 secretion system, as you can see. And the efficacy of the monoclonal antibody is preserved even when it is injected 4 hours after the onset of infection. So, could be possible to use that type of antibodies in human beings. We did a preliminary randomized control trial a few years ago using such a monoclonal antibody for the prevention of ventilator-associated pneumonia caused by Pseudomonas aeruginosa. So, we randomize patients colonized by Pseudomonas aeruginosa, not infected yet, colonized, to document the potential benefit of such a monoclonal antibody. At least it was possible to document a signal in favor of the new monoclonal antibody. Preliminary data, of course. It's also possible to target the LPS, you know. But unfortunately, it's, for the moment at least, it's only possible to target a very specific strain of Pseudomonas aeruginosa, the serotype O11. So a very specific population of microorganisms. So in clinical practice, it will be probably not very efficient, because before to inject the antibody, you need to be able to document that the patient is colonized and or infected by serotype O11 Pseudomonas aeruginosa, so which is not so easy. But in a preliminary study, we were also able to show at least some benefit, because compared to the predicted mortality in that observational study, it was possible to have a very high survival rate. Preliminary data once again. Now, it's also possible to build up a monoclonal antibody targeting two virulent factors, targeting the type 3 secretion system, as shown before, but also targeting the exopolysaccharide PSL at the surface of the bug. And as you may know, those exopolysaccharides are used by the bug to adhere, to aggregate to the cells from the host. So we are now targeting two virulent factors in order to treat or to prevent the infection caused by Pseudomonas aeruginosa. And we have also good animal data, as shown on the slide. As you can see, using the new B-specific monoclonal antibody, it was possible to improve the prophylaxis, the prevention of the disease in mice, and it was also possible as a therapy to show the benefit of such a monoclonal antibody. We are now conducting a phase 2 study investigating the potential usefulness of using such a monoclonal antibody, bispecific monoclonal antibody, in patients colonized by Pseudomonas aeruginosa, not infected, for the prevention of pneumonia in mechanically ventilated patients. So we have starting the inclusion into the study, and we will see, of course. It's also possible to use a different monoclonal antibody, and there is also an ongoing study done by Aridis evaluating the potential of such a monoclonal antibody. As I said before, it's possible to design specific monoclonal antibodies for targeting specific bugs. And it's now possible, for example, to target the alpha toxin produced by Staphylococcus aureus. As you know, the alpha toxin is one of the most powerful toxins used by Staphylococcus aureus to kill the cells from the host. So we will see once again, but once again, very good animal data showing the efficacy of this new monoclonal antibody, but also maybe the possibility using this monoclonal antibody to reduce the bacterial burden into the lung in mice infected by Staphylococcus aureus, and also in the kidney. So avoiding the dissemination of the bug into the body on the mice. This is interesting, of course, because targeting the virulent factors, you are not killing the bug, and you are effective, efficient, whatever the resistance to antibiotics. Using that type of monoclonal antibodies, it's of course possible to target methicillin susceptible cocci, but also MRSA. So you are now effective against bugs, whatever the resistance to antibiotics. And there is an ongoing study evaluating the potential utility of such a monoclonal antibody, targeting patients colonized under mechanical ventilation by Staphylococcus aureus. Two other pharmas are doing practically the same type of studies using different antibody. There is one study from Aridis and another ongoing from Arsanis, as you can see. Another possibility would be to use bacteriophages. As you know, bacteriophages are viruses that infect bacteria, in fact. And the beauty of this new weapon is that when a bacteriophage invade a bacterium, it replicates inside the bacterium, kill the bug, and then it's available to infect other microorganisms, and therefore amplifying its mode of action. But there are a lot of potential drawbacks. First, you need to get a lytic phage, and also you need to have the very specific bacteriophage for the bug you are targeting. And it's also possible for the bug to become very rapidly resistant to the bacteriophage. So not so easy to use probably at the bedside, but we will see. We have some very interesting data in mouse model showing the efficacy of bacteriophage for improving the outcome of mice infected by Pseudomonas aeruginosa, for example. So at the moment, because of the better understanding we have of the factors used by microorganisms to target the host, it could be possible to change our strategy, going from a terminator strategy to maybe a less violent strategy, targeting the virulent factors. And interestingly, it could be the way to go for preventing infection or for treating severe infection. But be careful. This is only the first steps we are making on a very long and difficult road. As you know, there are many possibilities for microorganisms to escape. They are using a lot of different virulent factors to target human beings. So we will see. Thanks a lot for the attention.
Well, thank you. That was very stimulating. I know the hour's late, but we could take a couple of quick questions. Are there any pressing questions from the audience? Jean, let me ask you, I think it's very exciting to talk about the monoclonal antibodies, but the trials, particularly for prevention, seem like they would be prohibitively expensive to develop. And then the use of these products seems very expensive as well.
Yeah.
So is this feasible, or is this just a good idea that maybe will never quite get off the ground?
We will see. It's impossible for the moment to say anything. Of course, before to use such a monoclonal antibodies, because not possible to administer the new monoclonal antibody to everybody in the ICU. So you need to be able to identify the most relevant patient, I would say. So the way to go could be to use the new technologies for identifying patients colonized by a specific virus. And as you know, Mike, it's now possible to do that. For example, using the Cepheid machine or whatever the machine you will use, it's possible to very rapidly identify patients colonized by MRSA, MSSA, Pseudomonas aeruginosa, and so on. So it could be the way to go.
Okay.
But yes.
A hopeful answer. Carlos, for you, do you see the same type of question? Do you see the applicability of aerosolized antibiotic for only refractory pneumonia that's not getting better with anything, or do you see it as routine adjunctive therapy that, if the data are positive, should be used for all people with VAP?
Well, what I think is that we need the clinical trials to see, first of all, if it is effective. And then, depends. In Latin America, in Argentina, like in Brazil, what we are seeing, where you are seeing now in your ICU, who have seen it 10 years ago. So the problem of the resistance is much worse than in the developed countries. But you are looking at what is going to happen in your countries in the future. So now the problem that they exist in much of the ICUs is that we have by sure 90% of resistance, and if we have this weapon, I don't know how to select in which patients to
Are you
use
using it now routinely or not?
No, we are not using it out because they don't have the trial that is supporting this, no?
Well, I want to thank our speakers. I want to thank you all for being here, and hopefully we will be able to continue future discussions about new ways to deal with this very difficult problem. Thank you again.